Introduction. Welcome to today's lecture on Krebs cycle regulation. In this session, we will delve into the various mechanisms that control and influence the Krebs cycle, also known as the citric acid cycle or tricarboxylic acid cycle, TCA cycle. The Krebs cycle is a central metabolic pathway that plays a crucial role in the generation of energy and the production of important intermediates for various biosynthetic processes within our cells. Now let's explore the 12 different aspects of Krebs cycle regulation in detail. The Krebs cycle is a series of chemical reactions that occur in the mitochondria of eukaryotic cells, efficiently oxidizing acetyl-CoA derived from carbohydrates, lipids, and proteins. It is regulated at multiple levels to maintain homeostasis and ensure optimal energy production. Regulation occurs through various mechanisms such as substrate availability, allosteric effectors, hormonal signals, and enzyme activity modulation. The regulation of the Krebs cycle includes control over the availability of substrates required for its proper functioning. Key metabolites, such as pyruvate, acetyl-CoA, and NAD+, act as central regulators. The availability of these substrates is influenced by factors like nutrient intake, cellular metabolic demands, and the activity of upstream metabolic pathways like glycolysis and fatty acid oxidation. Allosteric effectors play a crucial role in modulating the activity of key enzymes within the Krebs cycle. Enzymes like citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase are subject to allosteric regulation by endogenous molecules such as ATP, ADP, NAD, and succinyl-CoA. These effectors can enhance or inhibit enzyme activity, depending on the cellular energy status and metabolic demands. Hormones such as insulin, glucagon, and cortisol also contribute to the regulation of the Krebs cycle. These hormones exert their effects through signal transduction pathways that modulate enzyme activity and intracellular signaling cascades. For example, insulin promotes glucose uptake and activates key Krebs cycle enzymes, while glucagon stimulates processes like gluconeogenesis to provide substrates for the cycle. Pyruvate, derived from glycolysis, is a vital precursor for the Krebs cycle. Its entry into the cycle is subject to regulation by pyruvate dehydrogenase, PDH, which converts pyruvate to acetyl-CoA. PDH is regulated by phosphorylation and dephosphorylation events mediated by pyruvate dehydrogenase kinases and phosphatases, respectively. These enzymes respond to factors like nutrient availability, hormonal signals, and the energy state of the cell. The NAD/NAD+ ratio serves as an essential intracellular indicator of the cell's energy status. High levels of NAD can inhibit Krebs cycle enzymes, such as isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase, while low NAD levels promote their activity. This regulation acts as a feedback mechanism to maintain the balance between energy production and metabolic intermediates required for biosynthesis. Reactive oxygen species, ROS, are natural byproducts of cellular respiration and can accumulate under conditions of oxidative stress. ROS can directly inhibit Krebs cycle enzymes, causing impaired energy production and compromising cellular health. Cells have developed defense mechanisms, such as antioxidant enzymes, to mitigate ROS-induced damage and maintain the efficient functioning of the cycle. Isocitrate dehydrogenase, IDH, is a key regulatory enzyme in the Krebs cycle. IDH activity can be modulated by multiple mechanisms, including allosteric regulation by NAD+, ATP, and ADP. Additionally, IDH is subject to post-translational modifications such as phosphorylation, which further affects enzyme activity. The regulation of IDH ensures the precise control of alpha-ketoglutarate production and NAD/NAD+ ratios. Similar to IDH, alpha-ketoglutarate dehydrogenase, alpha-KGDH, is another critical enzyme in the Krebs cycle. Its activity is tightly regulated by NAD/NAD+ ratios and phosphorylation events, with various kinases and phosphatases acting as modulators. Different mechanisms and signaling molecules dynamically control alpha-KGDH activity to maintain an appropriate metabolic flux and adapt to changing cellular demands. Energy sensors and signaling molecules, such as AMP-activated protein kinase, AMPK, and sirtuins, play important roles in Krebs cycle regulation. These molecules sense and respond to cellular energy levels and metabolic demands, modulating enzyme activity and gene expression to optimize energy production, metabolic flexibility, and maintain cellular homeostasis. The regulation of the Krebs cycle can vary across different tissues and organs to fulfill specific metabolic requirements. For example, the upregulation of certain enzymes in adipose tissue supports lipogenesis, while in muscle, it promotes energy production and flux towards ATP synthesis. Tissue-specific regulation ensures the utilization of metabolic intermediates in a context-dependent manner. Genetic mutations in Krebs cycle enzymes can lead to metabolic disorders characterized by impaired energy metabolism and dysregulated biosynthetic pathways. Examples include mutations in succinate dehydrogenase, fumarate hydratus, and isocitrate dehydrogenase, which have been linked to disorders like mitochondrial diseases, hereditary paragangliomas, and cancer. In conclusion, the Krebs cycle is tightly regulated through a complex interplay of various mechanisms. Substrate availability, allosteric effectors, hormonal signals, and enzymatic modulation ensure the precise control of metabolic flux and energy production. Dysregulation of these regulatory mechanisms can contribute to the development of various metabolic disorders. Understanding the intricate regulation of the Krebs cycle expands our knowledge of cellular metabolism and provides potential targets for therapeutic interventions.